Hi everyone, welcome back to the EBI Seminar Series. Continuing our theme, Biomass to Breakthrough, today we're joined by Professor Chris Rao, Chemical and Biomolecular Engineering Professor at the University of Illinois Urbana-Champaign and Deputy Director of the EBI. In this session, we'll hear about his work on microbial engineering for the production of biofuels and other products. Thank you for watching and enjoy. Hi Chris, how are you doing today?
I'm doing great.
Awesome. To start off, what does it mean to engineer bacteria or yeast for biofuel production?
So the basic idea in engineering is that if you think about all the different chemical reactions that are going on in the cell, those are catalyzed by proteins, which we call enzymes. And the ability of a microorganism to grow on a certain kind of chemical or produce a certain kind of chemical is dependent on the enzymes present in the cell. And those enzymes are encoded by genes in the chromosome of the organism. When we engineer a microorganism, what we're doing is we're adding genes to that microorganism that provide new enzymes and new possible chemical reactions that that cell can catalyze, or that the enzyme can catalyze. We can also take out genes in that organism to prevent it from producing certain kinds of, or catalyzing certain kinds of chemical reactions. So an example might be Saccharomyces. So baker's yeast, and this is a microorganism we use to make alcoholic beverages, also used to make bread, but let's focus on the alcoholic beverages. It produces ethanol from sugar. It's a great hardy organism to work with. We have tons of experience doing fermentations or ethanol fermentations, but maybe we want Saccharomyces or the yeast not to make ethanol and say produce some other chemical, maybe butanol. So a 4-carbon alcohol versus a 2-carbon alcohol. So what we would do is we'd introduce the enzymes into Saccharomyces that would enable it to produce butanol. So that would be the addition of genes to it that encode enzymes. Simultaneously, we'd probably want to take out the enzymes that are producing ethanol. So the cell's not trying to produce both ethanol and butanol at the same time. So you'd have competition. We would eliminate out the ethanol and only have the butanol part. So that's what we do by engineering. We introduce enzymes into an organism. We also take out native ones.
And expanding on that, could you give us some examples of some of the techniques scientists use to engineer bacteria or yeast?
Yeah, so there's lots of different technologies, and there's new ones coming, every year. But kind of the basic idea to introduce genes into an organism is we work with something called a plasmid. And this is a small circular piece of DNA that replicates inside of microorganisms. So you have plasmids in bacteria, you have plasmids in a lot of yeast. What you can do is you can pull out those plasmids from the microorganisms. You can isolate those plasmids. And then what you do is you kind of cut out pieces of those plasmids. So you might cut out a piece of DNA from that plasmid, and you introduce the gene of interest into that plasmid. So we cut the DNA with restriction enzymes. So that's kind of cutting, the piece out, and then we put a new piece in, and then we use a DNA ligase to kind of glue it together. And once we have that plasmid, which is easy to work with in the lab, right? It's fairly small. You can sequence it, in a couple days. Then what we do is we transform the bacteria. So we take the plasmid and we stick it back into the microorganism. And there's a host of techniques. Typically, what you do is one of the technologies is you do electroporation. Basically, you shock the cell, an electric field, or create an electric field that kind of causes the membrane to kind of come apart or form holes in the membrane, which allows the plasmid to get in. Because it's pretty small, it can kind of get in. Other techniques are you can do chemical transformation, where you kind of make the membrane in the cell permeable. You can also use something known as bacterial conjugation, or you can in that way, this is commonly used in a lot of bacteria. You have one bacteria that natively will try to stick DNA into another bacteria. You can think about sex among microorganisms. You co-opt that process, and you get one bacterium to take the plasmid of interest and stick it into the other bacterium. You can do the same thing with yeast as well. Yeah, so that's kind of introducing DNA in. To take out DNA, like if you want to take out an enzyme, probably the technology that everyone uses now is CRISPR-Cas9. I won't go into details on that. There's probably a million videos on this. One of the most remarkable technologies that I've seen. And it's not that old yet. basically every synthetic biology lab uses it. But basically, at the heart of it is CRISPR-Cas9 is a way to cut DNA. And you can cut DNA in the chromosome so you can pop out parts, kind of like cutting and pasting in a Word document.
And what kinds of feedstocks are used in this process?
Uh, so right now, the most common is sugar substrate. So in, say, the US, when we make ethanol, we make it from cornstarch. So you take the corn kernel, inside of it is starch, and starch is a polymer of glucose molecules. And so you can think about it as beads, like along a string, where each bead is a single glucose molecule, and the string is the glycosidic bond between the two glucose molecules. You add enzymes to it, typically an amylase that kind of depolymerizes or cuts all the strings between the individual glucose molecules. And then you feed it to Saccharomyces, which can grow on glucose, and then it'll produce ethanol. The other most common substrate is sugarcane juice. And this is sucrose, so no different than like natural sugar. It's a dimer, it's a glucose molecule and a fructose molecule. Both of those molecules Saccharomyces can use. Typically, you add invertase, which will cleave the glucose and fructose, and then the organism will take it in and produce ethanol or any other kind of chemical that microorganism might use. Now, there's been a lot of interest in moving away, particularly from corn, because that's also competes with food, and looking at other substrates. So if you think about corn itself, if you think about the corn plant, it's a fairly large plant that gets about 6 feet tall, right? And the corn starch is contained in, the head of the corn or the corn cob or kernel. I don't know what the exact terminology is, right? But that's a tiny part of the biomass. You have this huge plant otherwise. And so there's a lot of sugar contained within that plant, but it's contained within polysaccharides. And polysaccharides are just polymers of sugar connected in kind of long molecules. But it's kind of challenging liberating those sugars out because microorganisms just won't grow on the plant biomass. Or if they do, they grow really slowly. Think of how long it takes, grass to degrade in your front lawn or leaves to degrade. It can take months, years potentially, wood even longer. And so you go through a processing step where you first chemically treat the biomass, right? Typically, it's an acid or base done at high temperature, and you kind of blast apart the fibers within the plant. And so you kind of liberate the polysaccharides. Then you can treat it with enzymes. And then you get this kind of what's known as a hydrolysate. And it's a mixture primarily of glucose, which is a 6-carbon sugar, xylose, a 5-carbon sugar, mixture of organic acids. And then you can feed that to microorganisms. The challenge with that, of course, is that there's also inhibitors that are the byproduct of this kind of transformation procedure to take the plant and make the hydrolysate. And those can inhibit the growth of a lot of microorganisms. So there's been a lot of work on making microorganisms more tolerant to these hydrolysates. Some are naturally good at it, but most aren't. But there's also been other areas of innovation, like looking at using food waste as a substrate for kind of fermentation processes. My work in the EBI is focused on using natural gas as a substrate. So there's microorganisms known as methanotrophs that can grow on natural gas or methane. And here we're trying to take a C1 molecule, methane is just one carbon, and make multi-carbon chemicals that are much more valuable than methane. There's other interest in using plastic waste as a substrate. That's still kind of a new revolutionary idea. But so I think, basically anything a living organism can grow on or potentially can grow on could be used as a substrate. But I've just listed the main one are, plant-derived substrates.
What qualities make these feedstocks, such as cornstarch and sugarcane juice, suitable for feeding these microorganisms?
Um, One is availability, that we can produce enough of it. If you think about like in the biofuel context, these numbers might be a little bit dated, but we use 1.4 billion liters of gasoline a day, right? That's a tremendous amount of material that we need to produce so we can drive our cars around, you know, have trucks move stuff, fly on airplanes. Um, and so the first thing is you want a lot of it. So you can make a lot of biomass, but it also has to be cheap. That's why there's a big interest in using kind of the leftover part of crops, the residue of crops, or using dedicated biomass crops such as grasses that, you can produce, are very productive on like a per acreage amount. So you want it cheap, but one of the challenges also is you have to be able to transport it. So, you have to be able to take all that biomass and transport it to kind of the production facility or your biofuel plant where you're going to do the conversion process. The challenge with biomass is it's not very dense. Think a pile of leaves, right? You can fill up a big bag of leaves, but it's not too heavy. And most of the weight is just water, right? So the actual weight of the leaves isn't that much. So that's kind of another factor is, one of the great is kind of the density. So one of the great things about corn is you have the corn kernels, right? So those get isolated basically at the farm, or the corn cobs get isolated at the farm. So you already have dense stuff. And so that's easy to truck around.
How do scientists choose which microbe to work with for a particular fuel or product?
Oh, this is a good question. I think there's two different lines of thought on it. So there's certain microorganisms that are just really easy to engineer. So E. coli and Saccharomyces are two examples. We've been studying these microorganisms for, I don't know, 50, 100 years. So we know a lot about them. The genetic tools are fantastic. So there's a group of synthetic biologists that work on these model organisms because if you're going to do any engineering, it's really easy to do. I take a slightly different perspective. What I try to do is take an organism that can already produce something close to what our target molecule is. So I primarily, most of my lab is looking at oil-related chemicals. So these are known as oleochemicals, which are typically derived from vegetable oil. And so we work with microorganisms that are really good at producing vegetable oil, and then we do genetic modifications to get that oil into a form that's ideal for the application we're looking at, being a chemical or fuel. So that's one example. The other example is the substrate. So when we do our methane work, we use specialized microbes called methanotrophs that naturally grow on methane. I think it'd be really hard to engineer a microorganism to grow on methane. I know there's a lot of efforts to do that by other researchers, but my perspective is, why not just start with something that already can grow on methane really well and then try to get it to produce a chemical. So we're focused on the downstream and taking advantage of the natural capabilities on the upstream. But the best thing you can have is you have a native organism that you isolate from the environment that already produces your chemical of interest from your substrate of interest. That's the best scenario, or requires very little genetic engineering.
Beyond fuels, what are some cool products that come from engineered bacteria or yeast? And are there any examples where we're already seeing these products in our everyday use?
Oh yeah, lots of them. If you've ever taken an antibiotic, that's from a microorganism. Penicillin, was isolated. That's a kind of classic one. What was that from? A fungus. So a lot of drugs are made from microorganisms. I think 50% or half of the top 100 top selling drugs are made by living organisms. Granted, they're not all made by microorganisms. A lot of them are made by mammalian cells that are used to produce things like antibodies or biologics. But we have a long history of, making drugs from living organisms. The next one would be obviously ethanol. It could be for an alcoholic beverage. It could be also ethanol that's used in gasoline as an additive. So that's probably the biggest thing we make by volume, be my guess, is ethanol. We produce a lot of ingredients in food from microorganisms: lactic acid, acetic acid, you can think of vinegar, citric acid would be another one. So these are kind of common food additives that are produced from microorganisms. Amino acids, which are used to supplement animal feed or even as a flavoring agent, like monosodium glutamate, are produced from microorganisms. In that case, they're typically not native microorganisms. There's been a little bit of engineering. A lot of it was done just by kind of, random chance, or you just try to find a mutant, because a lot of these technologies were developed before genetic engineering, but that's kind of a common product. Probably the best example in synthetic biology would be 1,3-propane diol, which is made by DuPont and Tate & Lyle. It's probably a 20-year-old process. And that involved a lot of engineering in E. coli. I know they're about to commercialize 1,4-butane diol. Both of these chemicals are used as precursors for plastics.
It's really cool. I didn't realize microbes were behind these products that we already use. How does microbial biofuel production compare to other processes for biomass conversion, such as HTL or pyrolysis?
Yeah, so HDL and pyrolysis basically what you're doing is you're taking kind of biomass and then you're heating up that biomass to high temperatures, high pressures. Often you add a little bit of catalyst, and you're what you're doing is you're kind of separating, pulling off the oxygens and the volatile components within the biomass, and you get an oil left over. So one of the challenges with biomass is you know, it's carbon rich and oxygen rich, whereas oil is carbon rich and oxygen lean. And so a lot of these processes are basically stripping off, the oxygen often in the form of water, and you're kind of getting this, insoluble or something that looks like an oil, like a pyrolysis oil. But it's like normal petroleum in the sense that it's a mixture of all different kinds of molecules together. And if you want to make gasoline from that or diesel fuel, you still have to refine that pyrolysis oil or HDL oil. Biology specific, or a lot more specific in the sense that if you're using, or producing microorganisms, it could be native, it can be engineered. It's taking a substrate like glucose or a hydrolysate or methane, and then it's primarily producing just one or maybe a mixture of related molecules. So you get a kind of cleaner, much more specialized product than you do with HTL or pyrolysis. You still have to separate out that molecule because you're growing it in a liquid medium. It's water, a mixture of salts. You have sugars in there, you have nitrogen sources, ammonia, but you're getting a much more specific molecule. And that's kind of the advantage of biology. It's fairly specific. Take a mess of compounds and it can make, one thing out of it. And so it's a little bit more specific, but you're still making those other things present.
So we've talked about the science and the applications, but moving from lab to large scale production is a different story. What are some of the biggest technical or economic challenges in scaling up microbial biofuel production?
Yeah, there's a bunch, and this has been a big challenge for the field. So we have certain processes that work very well. So corn ethanol, sugarcane ethanol in Brazil, those are great processes. But people, at least in the U.S., want to move away from corn ethanol just because you're competing with food. It's not probably the best process from an environmental perspective. So one is the challenge is producing a cheap substrate. So there's been a lot of work on different kinds of substrates. So what kind of plant should you use to make a biofuel? So right now we're using corn, but maybe it would be better to use a dedicated energy crop. And typically these are grasses that are perennial, so you don't have to reseed them every year. You just kind of mow the grasses. They tend to grow really tall, like 10 feet tall. So it's a much easier process to produce them. Then you have to actually transport that biomass. And that I already mentioned is a challenge because it's not very dense, right? And then you got to store, think of hay, like a lot of hay. And then you got to take that hay and convert it into the hydrolysate that you can feed in the fermentation. That hay is often very dirty and messy. So there's a lot of technical challenges on how to develop processes dealing with this kind of new kind of substrate. I think if you think about the fermentation and downstream, I think a lot of the core science is known. We can produce all different kinds of chemicals in the lab. We can produce them at high concentrations. One of the big challenges is scaling these up. There's just not a lot of expertise in running really large fermenters. So I mean 1 million liters versus, the 1 liter fermenters we run in the lab, or the 100 or 1,000 liter fermenters we run, in pilot scale plants. These are kind of laboratories where you start looking at the scale up of the process. So there's a lot to be learned on that. We don't have a lot of expertise. And building a biofuel plant's a lot of money. It's close to $1 billion. So you have to have a really good process to get investors to commit that kind of money to kind of build it up, and understanding that there's going to be a lot of teething problems, right? That we don't have a lot of the engineering expertise, and you kind of learn on the fly as you go.
Looking at the bigger picture, what role could synthetic biology play in the broader clean energy transition?
Oh, I think a lot of them. I think probably the most exciting to me is plant genetic engineering. I know GM food gets a bad rep, but the ability of synthetic biology to improve the productivity of plants is immense. Plants are, taking CO2 out of the atmosphere. It's the best way we have to reduce existing CO2 in the atmosphere. And so there's been a lot of work to engineer plants to be more efficient at photosynthesis and more efficient at CO2 sequestration. And so if you have better plants, that's kind of the starting point. And one of the challenges with a warming planet is that photosynthesis becomes less efficient, even though you have more CO2 available. And so you can use genetic engineering to make the plants more efficient. They can be more heat tolerant. With droughts and some of the problems with climate change, you can make the plants drought resistant, pest resistant. And so there's tremendous application on the plant side. I think at the microbial side where I live, we've gotten to the point where we've gotten really good at genetic engineering microorganisms. So we can read the DNA really well. And we're getting quite good at writing DNA or synthesizing chromosomes de novo. I like to tell my students that, so on the reading side, we can identify nouns really well. These would be the genes. We're also getting good at coming up with new words, right? So this is some of the evolution in protein engineering. I think AlphaFold, some of these AI programs using to predict structures of proteins that are really efficient at it is helping us a lot. The computational tools using AI are phenomenal in that space. They're only getting better. And we're starting to learn, what the verbs are in DNA and some of the grammar rules. We're not there yet, but each year we're getting better. So we're now we're getting to the point that we can design entirely new living organisms, ethics issues aside. That opens up all kinds of capabilities and the kind of chemicals that we can consume and produce. I think there's a lot of interesting work, say, in plastic degradation using microorganisms to break down plastics in the environment. There's a lot of work in environmental remediation, thinking about using engineered microorganisms to eat up the toxins, in our water and our soil. But then, if life can make it, we now have the ability to engineer microorganisms to make stuff, all kinds of new chemicals. So it's really just kind of your imagination is what's limiting you. So I think, as we get better, the technologies get better, the possibilities are just increasing. And the advantage of microorganisms is that the engineering cycle is so much faster. Plants, take months to grow or weeks to grow. Some of our microorganisms double every 30 minutes. So we have a much faster turnaround on the microbial side.
Well, Chris, thank you so much for joining us today and sharing your insights. It's been really interesting hearing about your research, and it sounds really promising, and we're excited to see how it continues to grow.
Well, thank you so much. I really enjoyed it.